All right, hello. Thanks everyone for joining us for this edition of the Soft Living Active and Adaptive Matter web series. So today it's a pleasure to have with us Dr. Eldad Afik from Caltech. He's currently a postdoctoral scholar and also at Caltech and also at the Howard Hughes Medical Institute, where he works with Professor Elliot Meyerowitz on spatio-temporal dynamics in organismal development. He's been awarded the Biology and Biological Engineering Divisional Fellowship in the Zuckerman STEM Leadership Program. He has a background in both physics and biology, quite aptly. So his BSc is in physics and biology from the Hebrew University, and following which he did his PhD research at the Weizmann Institute with Professor Victor Steinberg. And I'll let him take it away. And also just a quick reminder that we will have an informal discussion without recording at the end of the hour. And then at 10:15 Pacific time, we transition into the grad student only part of this discussion. So, and we also encourage questions during the talk. You may either just speak up or leave your questions in the chat, and we will periodically read them out to the speaker. With that, I will give Eldad the floor.
Thank you very much for this kind invitation, and thank you everyone for tuning in. It's very exciting to have these opportunities to share the work despite the distances and still some traveling limitations. As was said earlier, I'd be happy to take questions during the talk and have this as close to a room seminar type of discussion as we can. This work that we're going to discuss today is a joint work with Professor Elliot Meyerowitz at Caltech, who's the group leader, plant biology, developmental geneticist, and Tony Liu, who, when he joined us, was an undergrad at Caltech and joined us as part of a SURF program as a summer student project. As mentioned in the title, we're looking at self-organized waves, but the interest that drew our attention to this is the general perspective of living systems as systems that seem to behave as systems that follow an internal model of the external world, that they make predictions, that they collect information, and that they seem to anticipate what's going to happen and act accordingly. In our case, if I would throw a ball at you, you would send your hand to the place where the ball is expected to be and not in a random direction. And part of my personal motivation is to try to see to what extent can we push pure physical thinking into deciphering such behaviors. So going back to anticipatory behavior, one of the things that is common to many living systems is timekeeping, not my strength, and synchronization, both internally, that is coordination between different organs or different cells, between different elements in the population, if those bacteria, and with the external world, that is phase locking with daylight cycles, seasons, and so on. We found that this organism actually seems to predict or behaves in a way that predicts the day-night cycle, and this is going to be the main topic of this talk. The organism is the one that you see on this cover slide. The aquarium in which it's sitting right now is about 1 meter in width, and what you see in front of you are 4 individuals, and each one of these 4 individuals is considered a single cell. It's multinucleated. It has something that looks like differentiated organs, something that looks like a leaf, something that looks like a main stem, something that looks like roots. But the whole thing is considered a single cell from the point of view that there's no compartments dividing single nuclei. So as far as we know, every nucleus cannot control a microenvironment that is isolated by membranes from other nuclei. So in that sense, we consider it a single cell. So this specific project started by me wanting to track morphogenesis, the dynamics in which the organism regenerate and creates its morphology. What you can see here are the dark light in the background, the dark light cycles of the lab. So that's roughly 24 hours every switching of the lights. And you can see a single blade, which is about 1 centimeter, initiating a growth of a stolon, of a stem-like organ. And at the edges, you may be able to see that at the proximal regions, that the hue is changing or the amount of greenness is changing. And so this is the thing that drew our attention, and we were interested to know whether these type of waves, whether they are intrinsic, if they have an autonomous period, or whether they are driven by the external conditions, that these are the illumination conditions, and whether these are somehow coupled to dynamical states of morphogenesis. So whether there's any relationship between those waves that we're observing here and the dynamics of growth, of regeneration, of dictating which organs will show up and so on. The redistribution of greenness or is associated in the literature, is attributed to migrations of chloroplasts, and I might talk more about this later in the talk. It remained an open question whether these have a circadian nature, that is, do they follow a day-night cycle? The means that were accessible to those who did the experiments in the past were not time-lapse imaging, and the type of measurements that they've done were not permissive of addressing such questions. To address those questions, what we did is created an experimental system that comes to address challenges which are they're rather generic in complex systems. That is, how do we make a system that will be both reproducible and scalable? And specifically, it's a more common challenge in biology. And the idea was to start from as homogeneous conditions as we can, with multiple samples growing under the same conditions. So taking advantage of the stronger generative properties of this organism, we started from a single cell, single individual. We chopped it, made sushi from it, and used 1 centimeter segments of the stolon, place it in well plates, and let it regenerate. Nowadays, what I do is I let them to regenerate for about 6 weeks, and a single individual provides for about, that starts from 1 centimeter stolon, provides for about 20 to 30 wells after 6 weeks, give or take. The other component, the other experimental component, is this Raspberry Pi mini computer based imaging system that is mounted on this 3D printed model that was designed with the help of Alan Kornfeld, who was a high school student who did a project with us in summer of 2019. And the idea is quite simple, is to have a system that's scalable in the sense that we can make many copies of and wrap them separately, isolate them from the point of view of illumination, and use LED strips such that the Raspberry Pi computer, which is placed here, it's kind of like a third in size of a standard smartphone, is controlling both the camera, both taking measurements of temperature and humidity, and controls the light. So if we're going to look at how such a process looks, this is of course sped up. I'll go out of the presentation for a second. Maybe it will allow me to present it in full screen. No. sorry for this. So what you can see in front of you are A12 plates. I'll speed it up. It's not easy to identify when there's a change in the illumination conditions, and that has to do with the fact that the camera is set to adjust such that both when it's dark and when the lights are on, the imaging looks very similar. So the one sign is that when the transition is, there's a small flash where the camera adjusts, and the other thing is that you might be able to identify when it's darker that there's some graininess, but that's maybe challenging given this resolution over zoom, I mean. What you can see is that the samples that are grown are pulsing more or less at the same time. That's because all the samples in the same plate are under the same illumination conditions. What we're going to do next is we're going to place different plates under different illumination conditions and explore what the interaction with this external driving force. Now, I mentioned earlier that this greenness that we observe here, we attribute to the chloroplasts. And so for those of you who haven't had the chance to think of why plants are green, this is attributed to the material called chlorophyll that absorbs strongly in the blue and in the red. And so what we see is when there's a white light shown on green photosynthetic organisms, we see mostly the green that is scattered back or reflected back. So one interesting thing that you could do is take a green plant, place it in a dark place and shine a blue LED on it. And what you will see is a red spot, because suppressing all the other light, you will see actually the fluorescence. There's also a fluorescence in some parts of the red, not only absorption.
Okay, Eldad, I have a quick question. So you said this video has 8 plates and 8 samples in each, but only some of them end up growing. There's only a smaller fraction of all the 8 samples that you begin with seem to grow into fully developed organisms.
Okay, so just to clarify, when I say a plate, I'm thinking of this thing that we see on the left, and what we're looking here in the image, in the video itself, that's one plate with 8 wells.
Oh, okay, I see, okay.
And your comment about not all of them regenerating fully, so typically what we get is at least 2/3 regenerating within the first 4 weeks.
Okay.
Some of them don't regenerate as well beyond the first 4 weeks. And yes, we typically also have some that don't regenerate. At this point, since this is a protocol that we developed and we're not familiar with others that have done this, it's not clear whether, it could be that it's intrinsic to the organism, and we could also attribute it to the person who does it, which is me. So it could be my sloppiness that they don't always regenerate as well. But 2/3, I think, if you check in the plant world for when they do seeding and so on, it's quite not a bad situation to be at.
Okay, thank you.
But the question is in place. Yes, as you can see, they don't necessarily follow exactly the same trajectory in morphogenesis, in when organs appear, how many organs would appear, and so on. So that's part of the challenge. What we asked about is part of the challenges of this experiment and the analysis. For example, try to imagine if you had to analyze these movies when you're interested in the dynamics of the greenness. You can see that this is essentially it's an active scalar that does some non-trivial dynamics in space. The waves themselves are not manifested as a very simple dynamic, neither in space nor in time. So. Are there any other questions about this video or about the experimental system? Okay, what we're going to track in this talk are basically what happens on a single blade. So one thing that I mentioned is that we want to trace this greenness pulsation. So there will be some dimensionality reduction, and there are several motivations to do this dimensionality reduction. One is the conceptually, if you remember the talk from Caleb Wagner from 3 weeks ago, for example, the idea of trying to understand a fluid dynamic situation in the point of view of dynamical systems. So imagine that I could have a variable psi, and there will be a phase space that has to do with its conjugate momentum and some other attribute that has to do with morphogenesis, and that would be maybe a reduced version of this dynamical systems phase space. From the pragmatic point of view, it's just huge amount of data. The video that you just watched is about 8 weeks. This system generates about 400 gigabytes per month per plate. If you want to do several different conditions, you can imagine how much data there is to analyze here, and it's prohibitive to do them one by one by human, and all sorts of these computational challenges. So what we do is the following. As I said a minute ago, we're going to look as a sample. So every time series we're going to look at here comes from a single blade as a function of time, and this blade is growing also as a function of time. Given what I said earlier about the chloroplast, that might lead some of you to have a similar intuition to what led us, is to focus on the blue channel, which is shown here as heat maps. Right on the left, it's a blade that when the lights just turn on, and then about 6 hours later, you can see that same blade. The heat map is of the blue channel. So dark means our interpretation is absorption in the blue. Absorption in the blue we associate with high density of chlorophyll. But again, that specific statement is more of an interpretation. There's more work to be done to actually show this relationship. But if you want to have like the simplest explanation of what we're going to track in this talk is basically the darkness in the blue channel. The motivation is that essentially this is what's green in the image, what shows up as green in the RGB images. So back to the challenges, the image analysis and the computer vision side of things. Tony joined us for a summer project in 2020 when he was an undergrad here at Caltech, and I was very fortunate to have such a talented, ambitious student to work with me, to collaborate with me. What you can see is he took a quite simple and very efficient approach for us to have results rather quickly while we are also working on a more automated approach. And so the idea is to use tools from video processing, where you can see here the RGB image. Then, as I said, we look only at the blue channel. He applies a threshold on the blue channel, and what you see here is the binary version of all those that were darker than that threshold in the blue channel. And on the right is a pixel count as a function of time. Now, every stripe in the background, the gray and the white ones, represent 12 hours of dark and bright lights. When I say lights on, when I say dark, it's in average photon flux, which is about 200 times smaller than the lights on. So the lights, even when we call it dark or lights off, there is still some light for us to do imaging. Among the features which are interesting for me is the fact that, I'll speed this one up again. So among the features, at least while the blade is growing, which is what you can see here, if you would try to, just by looking at this data without having any knowledge, you might think that since this ebb starts before the lights are switching off and the flood starts before the lights are switching on, it might be that the sample is the one controlling the illumination conditions, because the behavior of the sample precedes the change in the external conditions, and that's what we refer to here as anticipatory behavior.
Eldad, I have
Yes.
another very basic question. What happens if you have constant illumination?
This is a great question of the first kind, the one I'm going to address in the next slide.
Great. Okay.
Thank you.
Yes.
Any other great questions? Okay, so to Suraj's question, we are going to do the same thing. So at the top strip, we are seeing the same sample we just looked at before. We're replacing here, instead of the RGB, we're showing you the blue channel inverted and rescaled, so they will all look more or less on the same level. In this middle stripe, we're going to switch from 12 hours on, 12 hours off to continuous off. Again, the off is just 200 times weaker. If we get to this at the end, I can show you that under what we consider dark, there's no growth, no morphogenesis. So at least from the point of view of chloroplast harvesting light, this is how this organism feeds in these conditions. It harvests photons and converts it to accessible energy biologically. Under these conditions, the dark is not being provided with enough light to grow. So from morphogenesis point of view, it's under starvation. The bottom strip is under continuous lights. What you can see here is as we switch the lights to the new conditions, sorry, one more thing to say is the time measured here at the bottom is in days. So this is 5 days, 10 days, 15 days, almost 2 weeks, from the time we changed the conditions. The reason why you see these blades showing up at different times, it's because these are different samples, each one from a different plate, and we're tracing blades. So I remind you, it's not that the sample started to exist at that point, it's the sample that we track, which is a blade that started at the point that it starts here. So what you can see as we switched the light to continuous dark, there's some transient here that lasts about 2 days. Actually, after these 2 days, there's a flip in the phase, but the oscillations persist. If we look at the 12 hours on, 12 hours off, you can see that the shape of the wave is asymmetric. During the daytime, it's more square, and during the flood is more round. If you look at the continuous dark, it looks much more sinusoidal, and it looks damped. So there are less, if we would have to guess, we would say, okay, here we have less higher harmonics involved. The higher harmonics are suppressed, and also the fundamental seems to be suppressed, but we'll see this promptly. For the continuous lights, I would dare say that at least at first glance, it looks chaotic. And it's not apparent, at least in first sight, that there's an intrinsic period that persists here. So any questions before we move to looking at the time series?
Can I ask another one?
Yes, please go ahead.
So this is all about essentially the density of chloroplasts, right? And this is the averaged green intensity across the entire blade.
That's our interpretation. Yeah.
Is there anything interesting that happens, at least roughly from the videos of the rasterized images? The one with the constant illumination looks like a much thinner blade but much longer, whereas when you have a lower illumination, it's much wider and stubbier. Is there any correlation as well with say the aspect ratio of the organ and growth rate, so something more spatial rather than just this integrated.
So I have two interpretations for the question. Please refine if I'm not meeting either. So my two interpretations is, one is, if you're asking about this specific video, then it's a specific realization, and the blade shape at until time 0 here is dictated by 12 hours on, 12 hours off. So the specific, everything that you see up to this point in time has nothing to do, sorry, until this point in time is not due to the change in the illumination conditions. It's just after this point that we change the illumination conditions.
Agreed.
The other thing about how the pulses take place, yes, I think it's interesting to ask questions about the spatio-temporal dynamics of those waves. The patterns that you see in the threshold in the binary version are not complete. It's not a complete description. There are pulses that happen in space, and specifically when you just look at the sum of how many pixels are beneath that threshold, we get rid of all that information. The reason to do that is it's a coarse graining that gave us insights into the system, and there's now more room to get into the complexity. But I'm not going to touch it at this talk. It's an ongoing process. And I'm happy to have more of this in the discussion. Like the specific, if you have specific ideas, then yes, please follow up in the discussion, because these are interesting questions to me. I think there's another question.
It's, do you know if the chloroplasts are dying off and regrowing, or are they migrating?
No, I don't have an answer to this, but I'll show later a reason why we think they are migrating. It doesn't exclude the possibility of dying off and being regenerated, but we have reasons to believe that there's a mass migration. Thank you. Okay, so this is a time for a quick challenge, and I don't know how we're going to do this over Zoom, but you are welcome to try to guess. Each panel here comes from a different plate. Some of them were under the same conditions. If we have any type of polling or hand raising or something like this, that would be great, but if not, then we'll just do this silently, each person to himself, and but you, as far as I'm concerned, you're welcome to interact if it works for you. So, given what we saw in the movie, any guesses whether this one would be what we think of as the reference, that is 12 hours on, 12 hours off continuously, even after this switching line that might be just artificial, and other. So is this a control? Is this a transition to continuous dark? Is this a transition to continuous light? We can try maybe raising hands if you think this is a control.
Wait, I have a question about your question. So there are multiple colored curves here. Like, are they all the same protocol, or they're different?
Each panel is the same protocol. These are samples coming from the same plate, not the same well. So these are replicas in the same plate. There are maybe other plates that have been under the same conditions in this plot here.
Okay, thanks.
Okay, so let's start. So this one is the control, and one of the ways to see this is by the persistence of the waves, and it might be challenging to see over the Zoom, but basically they are relatively square waves. If you had to guess another one here, which is a reference, or this what we refer to as the control, it's this one. Now, one of the ways to guess this is actually by elimination, and the elimination is to find those that show sinusoidal damped dynamics. So those of you who can follow despite the resolution over zoom, this is one striking. The friend of his did not grow as much, which is this one. So we still have the two transitions to continuous high. This is one, which, as I remind you from before, they seem more chaotic. And this one, which seems damped, you could still see some signs of things that is not just a damped harmonic behavior or near harmonic behavior. The one that I haven't mentioned, haven't shown you before, is this one, which, if I had to guess without having any other knowledge, I would think it's continuous dark. This one is continuous half of the intensity of the light. So this is also continuous conditions. What you cannot see in this image, and we might get to it at the end, is that this one continued to grow, unlike the two continuous dark. So from the point of view of the dynamics of these pulses, the way we do this coarse graining, it looks fairly similar to the continuous dark. From a morphogenesis point of view, it grows. Okay. Okay. What we're going to do now is to quantify those statements about the dynamics of the pulses. And we're going to do this by looking at the spectral composition by applying discrete Fourier transform and looking at the pulse spectra. The two controls, I'm sorry this is not organized in a convenient way for you, but the two controls are this one and this one. And what characterizes the control, I'll just mention here, the frequencies are in days. So this is, and it's represented on the log-log plot. So this is once per day, 10 times per day, 100 times per day. We can see in all the panels, there's a pronounced peak near the once per day. The controls show higher harmonics at integer frequencies of that pronounced peak. So there's once per day, twice per day, 3 times per day, and so on. Now, if we think of a square wave, we should expect odd integer multiples of the fundamental. We see here also the even ones. It's not easy to see here, but the even ones are slightly suppressed compared to the odd ones. But we still should expect the even ones because if you remember from before, this is not a purely square wave. In addition to the growth, there's also a difference between the top and the bottom, the flood and the ebb behavior, or the day and the night behavior. Okay, I'm switching to focusing on the continuous dark. The two continuous dark are this one and this one. The peak at once per day is pronounced. I'll show you in a second a blow up on these. And these are the two continuous lights on. Still, there's some peak at the once per day. To clarify, because the dynamics is non-stationary, we look at the power spectra only 2 days after the transition of the illumination conditions, after a point that at least we should be able to consider it to some extent stationary. One interesting feature here is how smeared the amplitudes are across the different harmonics, which is another signature of a chaotic behavior. Again, this is the continuous, the continuous high or continuous light. Okay. So when we focus on the continuous dark, look only at those that have the dots on, only the lines that have the dots on. One of the things you will see that it is shifted a bit to the right or to higher frequencies compared to the once per day. So it's slightly faster in frequency than the once per day. It's about 1.2 per day. The rest of the harmonics, you'll have to trust me on this, are less pronounced when we compare it to the reference, which is running under 1212. If we look at the continuous high, Again, look only at the data that has, so in the background you see those that don't have the dots are the reference, are those that kept running at 1212, where you can see those pronounced peaks at the integer harmonics. For the continuous high, there is still some memory of this once per day. It's closer to the once per day than in the continuous dark, and there's something that looks chaotic, at least from the point of view of the power spectrum. This is another example of the continuous lights on. You can see that there are some bumps here, but some other from the same plate are flatter again. And anyway, it is smeared compared to what we saw in the 1212. And the peak again is closer to the once per day, or if anything, it's maybe slightly shifted to slightly slower frequencies. Again, this sample for since 2 days hasn't received any cues from the environment of external changes in the external of a day-night cycle. So any questions up to here?
Yeah,
Okay.
I have a question.
Yep.
In terms of the frequency response, I guess you're suggesting that some of it, at a higher frequency could be chaotic, but could it also just be that the way you're sampling it, and this is really noise in terms of either aliasing or just sampling, or that you actually are looking at a signal that is not representative of something that's process related?
It's a great question. Of the second kind, the kind I don't have an answer to. I'd love to have your input about this towards the end. We need to still remember that when we look at the cases where we have the 12-12 continuous, we don't see this. We can see the pronounced peaks going all the way beyond the 10 per day. So we need to understand why this idea of noise would apply only in the case of the continuous high. But maybe there is a way to understand this way. It's not straightforward for me, so that's why I would appreciate your input.
I'm just kind of thinking about it in general, in terms of signal processing. Generically, you could have a window where you imposed it a priori, so then you don't really analyze or do frequency spectrum of something that's outside the bandwidth. If you follow what I'm saying, you could do it optically or.
Yeah, so we applied a Gaussian window, which width was about 4/5 of a day, and the window, so that was the scale of the Gaussian, and the window as a whole is 6 sigmas, 6 times this scale. So it covers more than 5 days. Okay. And we do oversampling here in the sense that there's zero padding to achieve 1/10 per day resolution in frequency, but you could say that this is artificial, this 1/10, because that's more of a and regarding the averaging of a noise, then this is using the Welch algorithm. So basically, we have more than 5 days or 6 days to average over. So we cut the signal and average over parts of it to kind of suppress the noise further. But again, I'm happy to have more comments, maybe in the discussion.
You actually answered my question, and I'm convinced that you took great care in the way you sampled it. Thank you.
Okay, it's good to know. Thank you. Okay, so we see that we have an intrinsic rhythm when we switch to continuous lights on or off. Now we're interested to see what happens if we drive it at very non-circadian rhythm. When I say very non, what I mean is that, again, at the top, you're seeing the 12-12. This is different data set. So this is a control from that same run, same incubator, same cut and regenerate procedure. This one will switch from 12-12 to 24-24. So that's a day of 48 hours. This one is going to switch to 6-6. So that's a 12 hours day. And this is a crazy planet that revolves every 3 hours. So it's an hour and a half. Among the motivations for this experiment was to try to see what happens when you push to the limits. Is it going to follow the external conditions at all, and how? And as a rough observation, it seems that the 24-24 seems to keep reacting to the surprise long night as a transient of the continuous dark, and then it gets light, so it gets out of that transient, and then it enters that transient again when the dark is too long, and so on. This is a very crude observation. When we look at the 6-6, you see those spikes here that seem to happen every time that we switch the lights on. But actually, they don't happen every time we switch the light on. They happen every second time we switch the light on. So these happen on a circadian period. Those spikes where my cursor is, for example, here and here and here, they happen every second transition. I think you could still see this despite the resolution that in addition to some wiggly that follows the lights on and off, you can still see something that looks circadian on top of that, or maybe close to circadian. It's hard to tell, I guess, just by eyeballing. So if there are no questions, sorry, about this video, I'll switch to look at the power spectrum.
How does the growth actually vary across these different protocols?
Great question of the first kind. You have many of these. You should keep asking. Let's get back to this at the end. Okay. But one thing that it needs further analysis, but okay, what I'm saying now is extremely preliminary, and we might have this conversation again in 2 months, and I'll tell you, no, I was wrong. But the observation is that there's a wavelength at the rim that seems to be associated with the changing of the lights when the sample is growing healthy.
Okay.
You could see at the blue one, for example. Now, I'm stretching the preliminary ones too much. I want to focus first on the things that are more sound.
Yes.
Okay.
And maybe this is not part of the main part of your talk, but how does the growth actually happen in this organism? So is it just at the growing tip of the blade, or does it happen everywhere? And what's the
mostly at the tips. Mostly at the tips. So the blade, the tip of the blade seems to be the place where there's growth. The tip of the stolon seems to be the place where there's growth. And typically, new organs appear only near growing regions.
Okay.
It's not only that, because when we start from the cut samples, they show also non-characteristic growth. But once they exceed some 5 centimeters or so, they start to show that type of characteristic that has been described in the literature. We can see things which are less typical in this system because it starts from a very small cut segment. Okay. I think there's another question.
Yes, what are the correlations between different parts of the same organism?
So we haven't explored it here. It's something that we were working on. The first approximation is that blades that are growing under the same conditions are passing in the same way. Now, the question that I would like to have an answer to is, are blades on the same individual more correlated than blades on different individuals when they are grown under the same condition, and this I don't have an answer for yet.
Oh, okay,
So
thank you.
I'm interested in this as well. I don't have an answer yet.
One quick question. So if you, in the previous slide, so if you return to the 24-hour cycle, is there a recovery of original rhythm?
A recovery of 24?
Yeah.
Okay, so that I think that would be easier to see here. So we have here the 12-12, the 24-24, the 6-6, and the 3 hours and a half, 3 hours and a half, sorry, 3 hours and a half, 3 hours and a half, and 6-6. So regarding your question, it's challenging to say. It seems to have a small peak here at once per day. I would say that with this one, we need to do the analysis a bit different if we want to resolve whether there is or there isn't. I don't think the way I did the analysis here can resolve the question of whether there is or there isn't a 48 hours, a half per day cycle. Because it goes back to the question that the gentleman asked earlier about how we do the power spectrum. I don't think the way we did it here can resolve whether there is a fundamental at once per 2 days. One of the interesting yeah, sorry.
Maybe maybe I misunderstood, but I'm maybe sort of continuing on what Niranjan just asked. Have you done experiments where, instead of just switching the protocol from, 12-12 cycle to a different, constant or different non-circadian forcing, but now switch it back to the 12-12 cycle after some period of time? And then is there some, how reversible is it? What's the recovery period in that sense?
Oh, sorry, I didn't understand this question. Okay, thank you for clarifying. No, this experiment hasn't been done yet.
Okay.
No.
Is there a sense of plasticity in how this entrainment happens, or is there any?
It's a good question of what is the entrainment and so on. I think maybe we'll go back to this in the discussion. Just bring it up back if you will at the discussion. Because of the time, I would like to complete at least the results part, and then maybe go into more of this both perspective discussions. So, I really appreciate the question, but I also understand that we have about 10 more minutes for the main part. So, the one that I did not expect to actually show any following of the external lights is the 1 hour and a half, 1 hour and a half. I expected I felt in my anticipation here. So this is the 1 hour and a half, 1 hour and a half. It shows a pronounced peak at once per day. It has a peak at the harmonic of that, and of the harmonic of that, and the harmonic of that, and then boom, at the 8 times per day. There's a pronounced peak which corresponds to 3 hours a day. If we look only at the pronounced peaks, they are the ones that correspond to the higher harmonic of the 8th time per day. So this is 16, 24. There's a small 32 here, 40, and it goes all the way up to it goes to 96 certainly. I mean, even at the 96, you can see 96 times per day. You can see a pronounced peak next to the smaller harmonics of the once per day. So for us, this was actually quite surprising that it can follow both the intrinsic and the external driving force and show a manifestation of both. Okay, I mentioned earlier that the continuous dark behaves a bit like a harmonic oscillator. So one could ask the question, what happens if we apply something which is like a small perturbations of in the frequency of the driving force, Now, one thing we have to keep in mind is that what I'm doing here is not a sinusoidal driving force, but a square wave, and that has to do from the point of view of simplifying the experiment. It makes the analysis more challenging, but if we go back to the Fourier, to the power spectrum, this is the reference, which is 1212. This is a 16th of a 24-hour cycle faster, a 16th slower, an 8th faster, an 8th slower, a 4th faster, that is, this is a 9 hours, and this one is a 15, that is a 4th slower than 24 hours. Sorry, 4th longer than 24 hours. It's a 30 hours day. Now, it was quite surprising for me that they actually follow the external condition in terms of moving their peak accordingly. So this is the kind of thing that I think where you could try to see that entrainment, and maybe in this type of experiment, it would be even more interesting to address what Niranjan asked earlier, where we could put it maybe under this 9 and then let go and see whether it goes back to the about 1.2 a day, which is the continuous dark, for example, or to the 1212. Okay, to summarize the main results that we looked at here, so switching from 1212 to steady illumination, we see that there's an autonomous circadian rhythm, that there seems to be 3 dynamical states that under the continuous daylight cycles of 50% duty cycle, we get the behavior that looks anticipatory, at least when the blade is young, and there's some asymmetry between the day behavior and the night behavior in terms of the dynamics of the observable. When we switch to continuous dark, the circadian is modulated to slightly faster frequency. The oscillations are damped, and the higher harmonics are less pronounced, so it looks a bit like a damped harmonic oscillator. The continuous light looks chaotic, but still we can see a peak near the once per day, and it's closer to once per day than in the case of the continuous dark. Under the perturbations which are far from 1212, we could still see in the power spectrum both the intrinsic rhythm and the extrinsic one, and the external drive, harmonics associated with the external drive, at least for the faster. For the slower, under the case of the 24-24, it requires more analysis. In our case, the experiment was too short to resolve that also. The perturbations at near 1212, which are kind of like a Q factor analysis, they show a circadian mode that could be modulated following the external conditions. Some open questions that we have in ongoing work, and some of these would address maybe some of the questions that came up in this conversation so far. So imagine I could write the dynamics for what we are looking at as a sum over discrete modes. Each one has an amplitude modulation and a phase modulation. So you should think of an amplitude, which is a function of time, and some sinusoidal function that has a phase, which is a function of time, not necessarily linear. So a purely sinusoidal function would be sine of something which is a phase which is linear in time, omega t. Here I'm thinking of a sinusoidal function that has some phase that is not necessarily a linear function of time, and that's what I'm thinking of as a frequency modulation. To resolve something like that, that reminds much more the type of sound waves that are happening here, that there's a frequency that maybe the fundamentals keeping the same, but when I say ki or ka or ku, there are different contents in the harmonics. If there are discrete modes and those are being reshaped like an AOE, something like this, given the external condition, that might be interesting also from the point of view of decomposing this behavior into the behavior of the circadian, what happens to higher harmonics and how they are related to the circadian. So the idea here was to apply a short time Fourier transform, or the Gabor transform, where basically every color column here is a power spectrum done based on a Gaussian centered at that time in time, but the Gaussian is wide enough for us to be able to do to pick up on the lower frequencies as well. Well, you can see here, for example, for the continuous dark, during the 12-12, you can see the once per day, twice per day, every such stripe is a harmonic. And when we switch the light to continuous dark, you can see most of them are fading out, and the continuous, and the once per day is being shifted slightly up. So that's what we saw also in the power spectrum. The question is, can we do this for arbitrary changes in the external conditions, or for questions like that was asked earlier, put it under 24-24, then switch back to the 12-12 and see those type of recoveries. But from the point of view of maybe there is a mode that we can think of as this is the circadian, and it could be phase modulated, it could be amplitude modulated, but that's kind of like an intrinsic something that maybe is a biochemical circuit inside or something else. And the rest might be related to some sort of metabolism happening with the chloroplast, which I remind you, the chloroplasts are the organelles that also harvest light and convert it into biologically accessible energy. Sorry, this is exactly the same plot, but now showing you the higher frequencies as well. Okay, from the microscopic point of view, we take the sample, we place it under confocal. In this case, it's a ARI scan, but similar type of. The bottom line, you get it. This is where we're looking at, where this small spot of light. This is a magnification of that area. And we're looking here at the window where when we shine blue light, we see the chloroplasts emitting in the red. So each individual that you see here is a chloroplast. And this movie is sped up 5 times. And the rate at which they are moving is about 85 microns per second. And this motion is actually meeting the ballpark of the waves that we see propagating a couple of centimeters over a couple of hours. So the orders of magnitude match. This experiment was not done with the idea that the light interferes. Actually, one of the reasons why I switched to the macroscopic imaging is realizing that it might be that when I shine light at it under the microscope, I'm actually interfering with the dynamics. But among the things that I think are interesting is that when you look at the motion, it doesn't look completely random. It looks like there are tracks. So these chloroplasts are probably following some micro filaments, cytoskeleton elements. And so it would be interesting to see what type of perturbations we can do to the underlying dynamics, and from point of view of other talks that were given here on active matter and organization of microtubules, for example. Like Alex's talk, for example, and postdoc I forgot from Christina Marchetti's group, where we could think of how this type of growth and the type of motion that happens maybe leads also to alignment that re-amplifies the directionality of the motion. Jacobs was one of the pioneers in the 60s to introduce this organism as a developmental biology organism in that time that was modern. So it was first introduced in the early 20th century, then again in the 60s. And then kind of slightly forgotten of. And he brought up the hypothesis that it is unclear what determines the directionality of regeneration, but given the vast size of the cell, cytoplasmic streaming is a candidate to create those types of polarity. You can think of, based on works by Goldstein, for example, in Kara, that a bulk motion of organelles, a collective motion of organelles on the boundary would create a moving boundary condition, and that could behave as a pump. From a metabolytic point of view, if, and this is a big if because I don't have a reason to think about this other than finding this hypothesis interesting, if the metabolism, the rate at which these organelles exchange material with their environment, is slower than the time for them to move, it could be that the type of metabolism that they did when they were spread over the blade keeps on going when they are concentrated at the base. And so that would create gradients based on that we deplete one region from the chloroplast and then we flood it again, and that could create some periodic type of polarity. So with that, I'm going back to the question of morphogenesis, and this is very preliminary. Again, a quiz. These are the cases where we switched from from 12-12 to either continuous dark or continuous light, or left the conditions the same, and one of them is the continuous half light. This is how the samples look the day before I switch the conditions. So at least from my perspective, I'm showing you 4 wells from each plate. From my perspective, they are roughly the same. Now, 4 weeks after switching the conditions, can you group them? Can you tell, if you cannot tell what were the conditions, can you tell which one came from the same illumination conditions? So some came from 12-12, that was not changed. Some came from switching to continuous dark. Some came from switching to continuous light. And one of them came from mid-light continuously. So again, a day before switching the conditions to the new ones, 4 weeks later. And again, given the time and the venue, I will start by saying that this one is paired with this one. These are the controls. They stayed at 12-12. If I give each and every one of you enough time, it will be easy for you to say that these two are the continuous dark. They hardly changed during those 4 weeks. These two are the continuous high. And there's a striking difference in the morphology. The blades are smaller, they're more frequent in space, and there's something that's challenging to see, but there seems to be a scar which might indicate some phototoxicity. This is the continuous mid. And some people, when I had the chance to do this interactively, were not sure when they grouped whether to group this one with this one, and this seemed different. So morphologically, there are enough people, say 3 or 4, that could be confused between the continuous mid and the 12-12. And I think this brings up an interesting hypothesis. The effect of the power, the average power, that is, these 3 got on average over a week the same photon flux on average. Because this is continuous mid, and these went through 12 hours on, 12 hours off. So the power, the photon flux for these on average is the same. For these two, the photon flux is doubled because they were continuous high. And this raises a question of maybe there is some sort of, and it's a wild conjecture at this point, maybe there is some phase diagram of dynamical states described. I'm picking 2 axes right now, but there are probably more. But the 2 axes I would think of is, one would be the frequency modulation, or say what is the phase modulation or the frequency of the external conditions, the average power photon flux, and there will be a stripe up here at too high photon flux that would lead, whatever would be the frequency at high enough photon flux, it will be phototoxic. At low enough photon flux, it will be starvation. And in between those dynamical states, starting from some damped oscillations, going through steady oscillations to chaotic behavior, And I don't know how the lines would look here, but we have some guesses based on what we saw of what happens when we play with the frequency. Okay, I'll skip the other types of perturbation that we're interested in. I'll just mention that there's the question of locality in space. So we could actually illuminate different blades on the same organism at different time zones and see the frustration in space. How local is the clock? And to conclude this, I will say the following, that we have a system that shows self-organization and anticipatory behavior and is amenable for quantitative research. Specifically in this work, we were able to resolve the pulses under different illumination protocols, something that was not available to previous research in the field about this organism, and that these waves exhibit both an intrinsic circadian-like oscillation and response to illumination protocol, and whether they are coupled to dynamical states of morphogenesis or whether there is no link, and the link that we see comes to do that both are affected by the light. This is for our future to resolve. So I'll conclude by thanking some funding sources. My collaborator, again, I was fortunate to have people like Tony and certainly Elliot Meyerowitz, my mentor in this group, and thanking you for all your questions and interest.
Yeah, so thank you so much, Eldad, for this talk and introducing us to this very exciting system. So let's all virtually applaud Eldad. And now is the time we switch to like an informal, unrecorded question and answer and discussion. So I will stop the recording now. And this will, we have like 7 or 8 minutes for